Alright guys, so here we go. Now we are continuing on. Last time we spoke about RNA processing. We discussed a bunch of things about how it's all capped, tailed, and spliced to make actual mature mRNA. And that was near page 41 of First Aid Biochem, where they talk about RNA regulation of gene expression and RNA processing in eukaryotes. The next page goes on to talk about RNA polymerases, where you have type 1, 2, and 3, right? So let's keep going with this. Number 12 here, RNA polymerases, and the same half of the page also discusses splicing, right? So we're going to be looking at all of that right here, and I have my a little drawing board here too now, so I can easily just go ahead and write as if I'm writing with a pen and paper, which really helps. So they tell you there's RNA polymerase 1, RNA polymerase 2, and 3, right? So we're right here. Okay, perfect. RNA 1, 2, and 3. That's what we're working with right now. So all of these things, you know, you're going to get tested on, but the majority of your questions are really going to be on type 2 right here. This guy is the most prominent, right? This is what you get tested on like 100% of the time, basically, because did not want to turn that into eraser. Okay, because that's kind of like the most important guy for your test purposes, at least. Oh, interesting. What did I click on? Became an eraser. How did I do it? Oh, here we go. Excuse me. First time actually using it. So they tell you in the book that type 1 RNA makes rRNA, type 2 makes mRNA, and type 3 makes tRNA. So you basically have an RMT kind of thing going on with the three guys. Each of these is very important, right? So you obviously need your rRNA, your ribosomal RNAs. Those guys are actually only found in the nucleolus. That's the most important thing about them, right? These guys are only found in the nucleolus. Okay, so that's your tiny thing, tiny really prominent thing inside the nucleus, inside the cell, right? So that really well-stained area, that's going to be where you're going to find your basophilic nucleolus, right? That's going to be often tested if they want to, not often tested, but if they want to ask you a question about RNA type 1, which are rRNA, then this is where you're going to find them. Right? So that's your rRNA. That's rRNA. And that's your type 1 RNA. So for number 2, this is your mRNA, right? Now mRNA are basically what we've been working with right now, the messenger RNA. That's the largest thing. It's huge, and that's the biggest type of RNA you're going to be working with your entire test. And we already talked all about mRNA, so there's not much else to say about that. Although it is the one that's going to get tested the most, because type 2 is everywhere. You know, you use it pretty much every single day to do what you need to do with making protein and all of that. UWORLD had another table where they told you that type 2 includes not just mRNA, but also small nuclear RNA and microRNA, right? So that was also prominent. There was also small nuclear RNA and micro miRNA. These are also type 2. Okay, the small nuclear RNA, those are involved in mRNA splicing, so that's also relevant for mRNA, of course. And then microRNA are actually responsible for mRNA silencing. MicroRNA are basically really, really tiny missiles that can go and destroy and degrade mRNA. So that leads to gene silencing, because if you don't have your mRNA, then you can't transcribe anything, right? And then the number 3 starts with a T, so you should just always think of this as tRNA for 3 RNA, right? The number 3 and T RNA both start with a T, so you'll never forget that. tRNA, we're going to see that that's the last thing left for our section of this chapter, where we're talking about the whole breakdown from DNA to RNA to tRNA to actually making a protein, right? The whole transcription and then translation. So tRNA is going to be the one that's helping you with that translation aspect of things, right? We've already done transcription. That was the one that helped us make the mRNA. Now we're going to do translation with tRNAs, right? So this is your group right here. That's the triad of things you're working with. Not much else. The book mentions 2 other little things that are side topics that you can get questions on, but you know, they're very straightforward, nothing crazy. There's alpha amanitin, and and then there's dactynomycin or actinomycin D. Now, alpha amanitin is not going to show up again in the book, so you do need to know it right here. It comes from Amanita phylloides, which is a death cap mushroom, and it basically inhibits type 2 RNA polymerase, right? So if it inhibits RNA polymerase 2, then you're going to imagine it's not going to allow you to make much of mRNA and so on, right? So this thing just stops RNA polymerase 2. RNA polymerase 2. I figure out how I can make the text a little less fat. RNAP2, like that. And that's basically it for that. The common format. Yeah, let's go with this. Maybe that's skinnier. Not really. Interesting. Oh, okay. Maybe that should do the job. Let's see. Yeah. Nope. Still became really fat. That is weird. How do you make the line thinner? It becomes fat as soon as it leaves the pen. Oh well, whatever. So we got the alpha mannitine. Now what is actinomycin D for? Actinomycin D, which is also, you can move the D to the front of the name, and it's also called dactynomycin. This thing inhibits RNA polymerase in both prokaryotes and eukaryotes, right? So it also inhibits, stops our guy RNA polymerase. So basically they both do the same thing, but alpha amanitin is only working on our RNA polymerase 2, and it's generally just going to be affecting eukaryotes, but actinomycin D also affects prokaryotes, right? So you can actually use this as, in a sense, an antibiotic. Let me see something. If I use the pencil here, it stays much thinner, but when I use it with this thing, it's always a really thick line. So maybe there's a setting in this where I'm supposed to make the line thinner. We'll figure it out later. All right, so there's that, and the other half of this page just shows this entire diagram where you're splicing your pre-mRNA and making it into mature RNA. So let me try to show you what you need to know here. So when you're looking at it in your book, on this page they have the splicing of mRNA with 3 steps. The primary transcript combines with small nuclear ribonuclear proteins, SNRPs, which of course, remember, small nuclear RNA RNPs we just talked about are RNA type 2, right? Type 2 RNA. So the SNRPs are gonna then go ahead and grab this thing. So in the book they show it to you as an entire piece, where in the beginning you have your exon right here, right, and then at the end you have your other exon. So remember, exons are what get expressed, right? Exons are what get expressed. In between this, all of this from here to here is an intron. Introns are intervening, right? And there can be multiple introns here or just one. It doesn't really matter. Now you see you have this thing called U1 SNRP that's attached right here, right? U1. And then you have a branching point in between here, which is your U2, right? Those are your 2 small nuclear ribonuclear proteins, and they're attaching in exactly this manner. This is what you're always going to be looking for. This mannerism is what you're going to get tested on, where they're going to give you huge list of nucleic patterns with all the amino acids, you know, GC, GCAA, whatever, all that stuff. And then you have to look at that entire gene sequence and figure out where exons and introns are going to be taken out. So this part right here is your 5' part, okay, and this part here is the 3' part. So your actual U1 attaches to the 5' part, and this specific area, if you look at it in your book, is the part that has the GU portion, right? So right here in this specific area, you have the GU portion, okay? So that's attached to your exon 1. And on this end, all the way over here, you have an AG. Okay, this AG is not relevant to the U2. U2 is just a branching point. This is just where things are going to twist around to break the intron off, right? But you have GU over here, and before the other exon starts, you have AG over there. So now, if you're ever given a long gene sequence where they're asking you where is there most likely an intron is going to be taken out, it's going to be anywhere where you could find this thing, right? So if you can find GU on one side and then a bunch of other things in between, and then AG on the other side, you can imagine that those are the two places where it's going to get cut off, and this everything in between is in intron, right? So if I show you a picture like this, for example, then you know where things are, right? So if you look at this image right here, you're basically seeing a whole intimidating looking list of gene sequences, right? This is one long gene sequence going all the way from 5' to 3', a list of codons. So now in this question, they can tell you that what would happen if I get rid of this G, and maybe this G becomes a C or something, right? So imagine that the capital letters are all your exons, and the lowercase letters are useless introns, right? So what would happen if I get rid of this G? Well, obviously, if you get rid of this G, you would lose this G right here, and now your splicing activity will get messed up. So you're going to mess up gene splicing. You won't be able to remove this intron, because this is good. This says GT, which would theoretically be GU on the RNA, right? So then when this becomes RNA, remember this is going to get imitated, and then that's going to become RNA, and based off of that, this is going to yield GU for you, and that GU is going to be good. The U1 is going to be able to attach right there. That's perfect. But then all the way over here, that's not going to work out too well, right? Similarly, remember that this is always pretty much exactly how things are going to be. You can also see the AG on this end and the GT on the other end, right? So these things are often going to be in the same manner. What we care about is this GU. This GU is associated with this GT. They're the same thing, right? This DNA is going to make a GU on the RNA, and this AG is this AG right here, which will be the same thing on the RNA, right? So you can tell this is DNA. Remember we talked about how DNA, there's a sense codon and an antisense codon. So if this is the thing you're trying to actually make protein out of, then you would actually be mimicking the antisense codon, and RNA makes sense. So your RNA is going to look exactly the same as this, except that all the T's will become U's, right? So there was a concept of sense and antisense, and that's something that shouldn't really be confusing. It's already been discussed in one of the earlier videos when we were up to that topic. So you definitely want to make sure you're watching this from the beginning to the end. I know it's taking forever to finish this, but once it is done, it will be the perfect full breakdown of everything you need to know specifically for step 1 without wasting time teaching you anything outside of step 1 stuff. All right, so that's generally it for this. Now how this is going to work is that your spliceosome is going to cut this off right here, right? It's going to make a bunch of folds, it's going to connect here, and then it's going to take this portion and cut this off here too, and you're going to get a little thing. You're going to get a little thing that you can just toss aside. And these two guys are going to get connected to each other, exon 1 and exon 2. Then you're going to have a tinier DNA basically, right? Or sorry, tinier RNA, tinier mRNA, right? Which is going to be the spliced version, and this is going to be your official mRNA, the mature mRNA, right? So that's all the splicing page talks about. The next page also goes on a little more, just talking about the same thing, introns versus exons. And on this page, they teach you something new regarding the idea of, what is this now? Topic 13. Okay. Introns versus exons. So in topic 13 here, they're going to teach you about how you can actually use the same DNA to make multiple different RNA and different proteins from the same DNA, that you don't need a specific sequence of DNA for each and every single protein you make. So they tell you in this page, they're showing you that there's multiple exons, E1, E2, E3 on this main DNA, right? They've labeled it all the way to E6, right? So there's multiple multiple exons on this DNA, and this is, I'm going to label it right here, DNA, right? So now, of course, you're going to take this, you're going to make it into hnRNA, which is a heterogeneous nuclear RNA that is not yet capped, spliced, and tailed, right? So that's going to be looking the same, basically. And then you're going to take that, and now you can splice this in multiple ways, right? So you have all your different exons, right? Your different exons. Each of these things is going to be basically giving you a part, right? So imagine that if you were building a human, right? So maybe part 1 gives you the head, part 2 gives you the hand, part 3 gives you the legs, stuff like that, right? You need all of them, generally speaking, in that example. But let's say that you're maybe making a, what are we making? Let's say we're making a car, and each of these are different parts of a car, right? So now some things are not in every single car. Like not every car has GPS, but some cars do have GPS. So imagine that number 2 is supposed to give you GPS, then I can put that in some of my new finalized spliced mRNAs. Maybe I'm going to combine just, I'll make a new mRNA over here. I'll make a new mRNA. And on this, I'm just going to put number 2, 4, and 6. Boom, there you go. That's my car right there. I can make another one where I'm just going to put 3, 4, and 7. Not 7, there is no 7. 3, 4, and 6. There you go. Okay. And so on. There's many combinations I can make of this. Many different permutations that I can come up with where I combine different things. And here you go. We made 3 different proteins, 3 different cars for my specific example, and so on. Right, so this is called alternative splicing. That's all this is. So this is a very critical topic that was, really mind-blowing the first time you learn about it, but then as you start studying more and more, you realize it's not really all that crazy. It's pretty simple and obvious, and a very nice design, that's for sure. Can't take away from the design aspect, right? So this is nice that you can do this. So just these 6 different exons can yield multiple different proteins. It's not that they can only make 6 proteins, right? Because you can combine them in different ways to make all kinds of different proteins. And that's all this alternative splicing topic is about. Right after that, they mentioned to you the microRNAs, which are type 2 RNA, right? And the microRNAs we said were, so there was the small nuclear RNAs, which we saw already were responsible for splicing, the SNRPs. And then microRNAs, these are responsible for silencing, right? So there's, if you go on YouTube and you look up videos for microRNA function, you can find really nice videos where they show you that microRNAs basically can go ahead and destroy destroy RNA before it makes protein, thereby getting rid of that translational activity, right? So RNA degradation is the key issue that microRNAs are going to help you with, and that's really all that was about. Very last topic to wrap this up for today is your tRNA. Now tRNA can be very confusing in questions, and you do not want to be confused about anything really when it comes to STEP. You should have as much knowledge as possible for every single topic so you can actually do the best possible. So they tell you in the books, tRNA have a structure of 75 to 90 nucleotides. They have a secondary structure of a clover leaf form. The anticodon is on the opposite side of the 3' amino acyl end, right? They're describing it to you all verbally, but the main thing is they can show you the structure very nicely in a picture, and they do that for you, right? So they have this structure where it's got the ACC for the acceptor stem, and then it comes down and you get your little cloverleaf form. You get this T arm on this side, right? There's a little variable arm that sticks out over here, goes down, you get your anticodon loop, right? And then it goes to the other side to give your D arm, D for dextro, and you should know that dextro means right. So this is on the right side, this is on the left side, right? So you have your T and D arm like that. And this is on the bottom here, you have your anticodon loop. Right, so you need to know what these words all mean, codon, anticodon, and stuff like that, because you're definitely going to get tested on all of that, so you don't want to mess any of it up. Now, the acceptor stem is really easy to remember with just the ACC, because ACC is how you start spelling the word accept, right? The acceptor stem is actually supposed to be read CCA. Right? So you want to make sure you get that, and the reason for that is because this part right here is your 5' part, and all the way up here is your 3' part. So you should always be reading it 5 to 3, right? So this is CCA, and the reason you would also remember that is because it's called can carry amino acids, right? CCA, can carry amino, and they tell you that in the book. This part over here is really important for basically carrying the amino acid relevant to the codon that's supposed to be here, right? So whatever codon is here, the amino acid is going to be brought to you attached over here. Right, so you can imagine from the whole table of codon to amino acid that there's going to be the correct attachment, and that's going to be very important. Correct attachment is going to get tested a lot. All right. So now if I tell you that I have attached, let me see, where's that table of the various things that are made? Well, actually, even if we can't find it, the one that I always remember is CCU. CCU gives you proline. Right? CCU gives you proline. So if we just use that, let's go back to the tRNA thing. CCU gives you proline. So remember 5' to 3', so it's 5' to 3', so it has to be so let's say I'm working with CCU. Let me put that for you guys here. CCU, this gives you proline. Okay, so now if you have a guy that's got proline up here, proline is attached to the acceptor stem up there, right? Then what would be written over here? Well, this is the codon. This is the codon going from 5 to 3 CCU. So then the opposite version of this would be C goes to G, C goes to G, and then U goes to A, right? So 5 prime AGG. So this is going to say AGG like that. Okay, AGG, this tRNA can, coming from 5 to 3, 5 prime AGG 3 prime, can attach to wherever I have my mRNA that says on it 5 prime CCU. And when this anticodon successfully attaches to this codon, it realizes that it needs to put the proline right here in this entire protein that's being made. Okay, so you can see how there can be mistakes that can occur, and it's very important to stop such mistakes from occurring, because the wobble position can first of all alter things. That's one issue that can occur. The other thing that can occur is that you might have the wrong protein attached to your tRNA to begin with, right? So what if proline wasn't actually the thing that's attached there? Then that would be a problem. Let me try and grab a picture here. Let's bring this over. Wow, my laptop server is blowing up. Trying to buy a new laptop. Sounds like it's about to take off to space with Elon Musk over here. And it started to slow down. Let me close some things. Alright. So you see how proline has multiple different things that can give you proline, right? So at least here, the wobble position kind of safeguards you from errors causing different proteins to be formed, right? So if by any chance, now, first of all, before we even actually talk about this, we discussed this way earlier, quite a while ago. What is the wobble position, right? Often, most people think that the wobble position is the thing on the mRNA, right? That third position on the mRNA, but it's actually not, right? So we all think that our wobble position, looking at this is all mRNA. These are mRNA things here, right? These are all mRNA things here, and of course, when numbers are not written, you assume it's 5 to 3, and it is. It's 5 to 3 CCU, CCC, CCA, CCG. Theoretically speaking, if all I give you are mRNA, then yes, the U, C, A, and G are clearly relevant to the wobble position, but it is not correct to really say that these are the wobble position. The actual wobble position is that A. Okay, the wobble position, and the book shows it to you right here, where they're talking about tRNA structure and charging. The wobble position is the first guy on the 5' side of the actual anticodon, right? So for our case scenario, our wobble position is the A over here in our anticodon, right? Anticodon goes from 5A. So if I was to write this correctly, or more standard, it would be 5AGG3, right? So this A is the actual wobble position, right? So now what can happen is you could have tRNAs where this A is something else. Well, it wouldn't matter what the A becomes, because all 4 possibilities yield proline. So you're pretty safeguarded from little wobble errors leading to issues when it comes to proline itself. Proline is very rarely misplaced or mischarged. Okay, it's not the one that ever really gets charged, because all 4 of these errors in that third position yield the same thing. All right, so that's the third position on the mRNA, but the real area of the position that we care about is the wobble position, which is the tRNA. The actual wobble position is on tRNA, and it's related to this portion on the mRNA, so that's what we always talk about. All right, so just a key conceptual little thing to make sure you have down. Other than this, the last thing they mention is that there's an enzyme that's actually responsible for attaching this proline here, right? That attachment is thanks to our guy aminoacyl tRNA synthetase. Aminoacyl tRNA synthetase. Okay, this is the guy that actually does the charging. So our charging is done by aminoacyl tRNA synthetase, ATS. Now, this charging never goes wrong. Aminoacyl tRNA synthetase actually also, as they tell you here for the charging, it also checks the accuracy. So they tell you in the book, aminoacyl tRNA synthetase uses ATP, one unique enzyme per respective amino acid, and binding of charged tRNA to the codon are responsible for the accuracy of amino acid selection. So there's a unique aminoacyl tRNA synthetase for each thing. So there's one for proline, there's going to be another one for all the other things, methionine, whatever you want to work with. So that being said, we have enough specificity to make sure there's no errors. It's because you have a unique one for each thing, they're going to make sure that you have the proline aminoacyl tRNA synthetase to attach proline here when it's the correct anticodon, AGG. Okay, and then every other guy is going to have their own thing. And it also scrutinizes the aminoacyl tRNA synthetase, matches an amino acid to the tRNA by scrutinizing the amino acid before and after it binds to the tRNA to make sure that the correct amino acid has been bound to the correct tRNA. If an incorrect amino acid is attached, the bond is hydrolyzed and it's going to be undone. You're going to remove whatever was attached here. A mischarged tRNA reads the usual codon but inserts the wrong amino acid. However, that's supposed to never happen, but UWorld does have a question where they tell you that they forced the wrong amino acid to be attached here. So maybe instead of proline, I'm going to attach methionine here. Now what's going to happen is everywhere there's a CCU on my mRNA, I'm going to have the correct tRNA, AGG is going to attach to it. However, I'm going to put the wrong amino acid. So everywhere there was supposed to be a proline, I'll have a methionine, right? Incorrect insertion. That never naturally occurs. We have enough safeguards to avoid such a problem. But in an experimental thing, in a question, you could do that and mess things up. Right? So that's all that's about. And that's basically tRNA in a nutshell. Other than that, you do have your T and D arms where you can get questions on. So that's not too bad. They tell you in the book that the T arm contains the TWC. That's what I've always called it. When I was studying for step 1, New York had a cable company called Time Warner Cable. It doesn't exist anymore. Now they became something else. But Time Warner Cable was always referred to as TWC, which is on the T arm, right? So you can see the W is actually pseudouridine, right? So ribethymidine, pseudouridine, and cytidine. These are not found anywhere. So if you ever have a question where they mention something has ribethymidine on it or something has pseudouridine on it, then it must be tRNA. That must be what they were talking about, okay? Because you'll never find pseudouridine on anything else besides tRNA. So you immediately, it's a giveaway for you that they're talking about tRNA in this question. And this thing is just a T arm for tether, right? It tethers. So they tell you in the question, in the book, that the T arm tethers tRNA molecule to the ribosome, which is the next thing we're going to talk about. So this is how it attaches to the ribosome. Okay, ribosomal attachment. So what is the D arm for? The D arm is actually going to detect the tRNA. Right? It's going to detect the tRNA, and by detecting the tRNA, it's going to be able to detects the tRNA by aminoacyl tRNA synthetase. So it's going to be allowing your dihydrouridine and aminoacyl tRNA synthetase to kind of work together and do the correct charging. That's all it's really for, right? Correct aminoacyl tRNA synthetase charging. So DR for charging, tRNA for ribosome, anticodon to attach to the mRNA, and the top is the acceptor stem that brings the amino acid. And that's your entire quad design, right? Four function design, top, bottom, left, right, for what a tRNA is going to allow you to do. Right? You can get all kinds of questions where they try to be tricky about it, but there really isn't anything tricky about it. Do not fall for weird, tricky shit that isn't actually tricky. You know, step 1 is always going to try to give you extra information on questions where they're really trying to kind of convince you into picking the wrong answer by telling you extra information. Don't fall for it. Don't listen to the extra information. Always pick and choose what information you care about, and getting good at that is what we do practice questions for. So we need to learn what we care about. Even when we get real questions, and by real questions I mean questions about patients, not questions about non-patient principle material. This is the principle. When you get real patient questions, and when you get real patients in the real world, you're going to get patients that come and tell you their life story. They may have come back just for a COPD exacerbation, but they'll tell you their life story. They'll tell you everything that's happening with their heart, their psyche, their kidneys, and so on. But you really don't care about everything, because if you start looking at everything, you start having too many differentials to worry about. You want to pick the most symptoms that go together, and then they're all going to pointing successfully at the right symptom, the right diagnosis, then you're good. If they have 5 symptoms for one diagnosis, but then they have this one more symptom that is completely against that diagnosis, then chances are it's not that diagnosis. And these 5 symptoms were thrown into the question just to confuse you, right? All the symptoms should match up to one diagnosis, and no symptom should deny that diagnosis. Oftentimes when we're on UWorld, we'll see a question where a lot of things are kind of sort of pointing towards lupus, maybe, right? Lupus nephritis, maybe it's a renal question. And then there's something they mentioned that sounds a little like glomerulonephritis too, and then you start thinking it must be glomerulonephritis, and you picked it, but you didn't realize that the question also mentioned that the patient had a malar rash, which definitely points more at lupus nephritis than pointing towards glomerulonephritis. No one gets a malar rash on their face with a butterfly design with glomerulonephritis. It's not a thing. So very important to make sure you can make such decisions and pick and choose the correct symptoms you actually care about. You don't always care about every single symptom a person has, and the very first sentence is very critical. The very first sentence of a question where they tell you a patient came in for COPD exacerbation, well, now you should really care about respiratory and renal things that are happening, respiratory and cardiac things that are happening, and not so much about any renal thing that might be happening to that patient. It's not really relevant to, or not, at least not highly and directly relevant to, their COPD exacerbation. Could be something else. You of course work with the question set, figure it out the best you can, and so on. But when you're practicing questions, really learn what symptoms should you not have focused on. So now for our last topic in this section is your protein synthesis. To wrap it up, they tell you a bunch of things here, right? And now we're using our rRNA, which is a type 1 RNA. And you have your ribosome. Now the ribosome is a pretty cool looking thing, right? We got a nice little guy we're working with. So it has 2 sections to it. It has the top part, and then it has a bottom part, right? And let me just make sure I'm not in the way. Okay, so that top part is going to be what they refer to as either 60 or 50, depending on what organism you're working with, and the bottom part has a smaller number, 40 or 30. All right, so those numbers are relevant to what organism you're working with, and they tell you that these 40 and 60, which are even numbers, the 8, remember 40, 60 gives you the ADS is even. They're just coming up with these numbers and telling you that the big even numbers are for your eukaryotes. So you have the 4, which is even, and the 6, which is even, right? So that's what they're talking about. The even numbers 4 and 6 go with the eukaryotes. The odd numbers 3 and 5 go with your prokaryotes. So that's what they're telling you. Right? The red colored numbers are what they're really talking about. Synthesis occurs from the N terminus to C terminus, and that's pretty easy as well. Nothing crazy about that. The real thing here is the concept of how these A, P, and E segments work. So you have an A segment, a P segment, and an E segment, right? That's your group of your three segments that you have to work with. I gotta figure out how to make the pen skinny when I use the board. I'm not sure why it makes it fat. We'll figure it out. I'll check after this session. So now the 3 letters A, P, and E at least help you figure out what's happening in each one. So the A section is the least busy one, okay? All that happens there is the incoming amino acid tRNA synthetase. The incoming amino acid tRNA, goes in there, the incoming one. The very, very first guy, okay? But to begin all of this, right, you can see in your book they have AUG. I don't know why they wrote AUG twice. You just need AUG once as the start codon, but okay. Actually no, AUG, CAU, right? That's what they have. So the AUG, you can see that it arrived, right? The very, very first start codon actually will arrive at the P site. Right? So AUG actually arrived at the P site. You can see that in the picture. And it's not just by chance that they're showing it to you like that. They tell you for initiation, eukaryotic initiation factors, identify the 5' cap, they assemble the 40S ribosomal subunit with the initiator tRNA, and then EIFs, initiation factors, the eukaryotic initiation factors, are released when the mRNA and the ribosomal 60S subunit assemble with the complex. This requires 1 GTP. Every step requires 1 GTP. So initiation, elongation, and termination each require 1 GTP. Now when you go to elongation, amino acyl tRNA binds to the A site. But then they tell you right here, except for the initiator methionine, the very first methionine, AUG, actually goes to the P site. Okay, so this AUG is your start codon, that goes to the P site. After that, you see they have CAU. CAU is the first real guy that's actually going to go to your, that's actually going to go into giving you something for your protein, for your final protein, right? The start codon doesn't really make anything, it's just a start codon, but CAG will make something, or excuse me, CAU, the way they have it in the book. So just remember that your start codon, your very first start codon, start AUG, goes to the P site. Okay, very important to keep that in mind. That part is something you can get a dumb question on, and you don't want to mess that up. After that, everything else that's coming, the whole thread of your entire mRNA that's arriving to the ribosome, will keep entering your A guy, and this is going to keep moving in this direction as the exit termination part, right? And this keeps going and keeps going and keeps going until eventually inside your A you get a stop codon, right? Once you get the stop codon in here, like UAA or UAG or something, then it's going to realize that that's a stop codon, and it's going to make these termination factors. They call it the eukaryotic release factors. So you had your eukaryotic initiation factors and eukaryotic release factors that will now release the protein that has been put together. And based off of the charges of the amino acids that you put together in that protein, it will assume a tertiary or quaternary structure and fold accordingly based off of whatever charges exist and whatever bonding occurs there. Again, not as complicated as it could be. They try to make this more complicated in questions than it really is, but this is the only key factor here that you need to understand. The rest of the things you just read and you know it, and you probably already learned it in med school, you know, or even in biology class back in high school or college. So not the most complicated thing. In first aid 2021, they made a more detailed drawing to show you how it all occurs. They're showing you elongation and all of that, and they continued to do a good job to remind you that AUG arrives in the P site. Everything else comes in the A site after that, and it all keeps moving in this direction. Also keep in mind that there are multiple ribosomes on one RNA. Multiple ribosomes working on the same RNA and just moving down and translating it into protein. So it's not like one RNA, one ribosome, one at a time kind of thing. Right? That would be too slow. Okay. And that's it. With that said, we're done with our molecular section, and then in the next video we'll continue on with our cellular section, right, which isn't that bad either. And I'm going to try to make more sessions more often now so I can get these videos done quicker for everyone that's preparing for step 1. Right? So thanks a lot for joining, guys. Keep working hard, and I'll see you guys again later.